Hey guys, hi. Okay, so here we are at another question. Now let us try to solve this. Now, let's begin. A student requires a blood transfusion. Genetic analysis shows that student's father is type B plus. So let's start with father. The father is type B plus and the mother is type A plus. So now the first moment you start reading the question, you are seeing that there is A and there is plus, B and there is plus. That means there are two systems that are going to be here at work, and that would make things complicated. You understand that people who are Rh positive, they have the Rh antigen, right? People who are Rh negative, they do not have the Rh antigen. But the problem is, if you give a negative individual the positive blood, his body or her body would develop the antibodies. Why? Because now the new antigen, the D antigen is coming. So body has to develop antibodies against it. So we do not try to do that. Okay. So that thing is later on removed here, that everybody is assuming all individuals here are Rh positive. That is later on removed, that everybody is Rh positive. So to a positive individual giving a positive blood, no problem. Okay. Now, a student requires a blood transfusion. Those are the parents, type B and type A. Now, what do we know about type B and type A? One allele must be IB, other allele must be IA, right? This much is for sure. The second allele, that is where the issue lies. The second allele could be IB or small I, and same case here, the second allele could be IA or small I. We don't know. Okay, now let's go ahead. Both are known to be heterozygous. Now they have mentioned both are known to be heterozygous. The moment they say they are known to be heterozygous, you understand that it is not going to be IB. You understand it is not going to be IA. That means both individuals are IA small I, IB small I. We pivot as per the question. Simple as that. A potential donor is identified. Now, what is their son? We don't know right now. But we understand what are the possibilities. The possibilities is A and I, right? That could be A. B and I, that could be B. A and B, it could be AB. I and I, that could be O. So as of now, the son could have any of these phenotypes. We don't know right now. We are not sure. So we are not sure, so we'll let it be. Okay. A potential donor is identified. While the donor's blood is unknown, the monozygotic twin is known. Now, monozygotic twin. What is a monozygotic twin? Mono, one. Zygote, the fusion of sperm and ovum. That means that this twin or these twins have come from a single zygote. So, single oocyte was fertilized by a single sperm. It formed the zygote and the zygote split before 8 cells. That is monozygotic. Okay, so this is monozygotic twins. Cool. And they are exactly same. Same oocyte, same sperm, hence the zygote is same. So is known to possess anti-B antibodies. Now this is where the confluence of genetics and the circulatory system lies, and you need to be very careful about it. The zygotic twin, the monozygotic twin, has anti-B antibodies. Now, what is the rule? I shall not have antibody. The rule is I shall not have antibody for which I have antigen. That is the rule. If antigen is present, corresponding antibody should not be present. Okay. Now, this guy has anti-B antibodies. If he has anti-B antibodies, and remember, we are talking about the donor right now. If he has anti-B antibodies, that means there is no antigen B. Sorry, antigen. If he has antigen, yeah, anti-B antibodies, there is no antigen B. And if antigen B is not there, that means most likely what will the person have? The person will have antigen A, right? Now, possess sorry, my bad. The question says is known to possess anti-B antibodies. Yeah, correct. So anti-B antibodies are present. That means no antigen B will be there, but antigen A will be there. Okay, so we should not have antibodies for which the antigen is there, but no anti-A antibodies. Makes sense? I have antigen A, I should not have antibodies anti-A. Perfect sense. So this confirms us, right? This confirms that the person has A antigen. That means the person is blood type A. This confirms the antigen is blood type A, and it also confirms it cannot be AB. Why it cannot be AB? Because no, if B were to be present, then the individual shouldn't have had anti-B antibodies, but he hasn't got. So the blood type A is confirmed, and we can also outrule O. Why? If O is there, meaning no antigen, meaning both antibodies should be there. So it's very, very clear that the blood type of the donor is A, but no anti-A antibodies. Assuming all individuals are Rh positive, what is the probability that the student can safely receive the blood transfusion from the donor? So the donor is blood type A. Now, who can A donate to? We easily understand A can donate to A and A can donate to AB. AB is a universal recipient and A can definitely take it from A. So we want to understand what is the possibility that the receiver, the person who needs, is A or AB, right? So this is what we want to find. What is the probability that the individual is either A or individual is either AB, right? This is a possibility they are asking. Now let us go back to our original thing. In our original thing, what were the possibilities of the sun? AAI, BBI, A and B, and O. So there were four possibilities. And what are our favorable possibilities? The favorable possibilities are A and B, right? Now here, what we'll simply do is favorable outcome is 2 and total outcomes is 4. Don't do this here. We'll just do a simple maths here, that favorable is 2, total is 4. So 2 by 4, so probability of A or AB is equal to favorable to total 4 is equal to 1 by 2. Simple as that. So our answer, what would be the answer? The answer would be 1 by 2. Okay. So such questions can easily come in the exam, and we should be okay. Thank you so much. We'll start with another video in the next class.